Hello everyone. I am going to present a short synopsis of our work on behalf of the Neurons United Consortium. So this presentation will be a little bit all over the place, so if you get lost, just feel free to stop me at any time if I get too carried away. So I'm going to start with a small historical background in neuroanatomy, and I'm going to say that unraveling the internal wiring diagram of the brain, regardless of its species, which we usually term the connectome, is of fundamental importance to neuroscience because this allows us essentially, it benefits both experimentalists and modelers. We can essentially build more realistic models of the brain and connect the experiments to the broader part of the picture. So as a prime example, actually, and also I want to say that over the past 20 years, we're kind of lucky to have so many advances in essentially neuroanatomy, data imaging and acquisition pipelines, as well as big data analysis and modeling workflows. So as a prime example, we'll go to 2014 and we have the study of Fromeau et al. That released this big data set that contained 500 anterograde tracing experiments performed on the mouse brain, which, for those who don't know, essentially is a bulk way of infecting a huge population of neurons, and then the infection is carried downstream through the axons and the terminals, and then with imaging you can essentially visualize and quantify densities of axons spread across the cortex or other areas, which after proper processing gives what we say the mesoconnectome, which is connectivity at the mesoscale level of anatomically distinct brain areas and the densities of projections between them. So this made the mouse, in my opinion, the species offering the finest trade-off between coverage and resolution in structural connectomics. And then we had a follow-up from 2019 from Harris et al. That released a similarly massive data set, this time performed on crystalline transgenic mice instead of wild type, which essentially effectively allowed them to target particular populations of cells that targeted, for example, the thalamus or the cortex from the other hemisphere, or going to the subcortical areas. But there are some limitations that I will focus as follows soon. So parallel to that, we have the release of the first full three-dimensional atlas of the mouse brain, which came from the Allen Institute, that provided the labeling and the parcellation of approximately 1,000 fine-grained sub-areas and nuclei of the brain. Both hemispheres came from the averaging of thousands of brains together. And this, why is it important? Because if you're a neuroanatomist and you have obtained your own imaging data, you can now register them to a 3D space and assess their spatial context and visualize them in 3D, as well as overlay them with data obtained from other modalities or the same modality, thus setting the fundamental basis for data fusion. So the limitation with the crystallines or the wild-type experiments is that even that way, even if you're able to target a particular population of cells, it's still not possible to track down the particular cell type that your cell type projects to. However, there is a beginning of a new trend, and this is something that maybe we could start talking about, which started in 2019. And as mentioned before, starting with the mouse-light project, we have this new trend on utilizing massive high-throughput industrial approaches to essentially fully reconstruct long-range projecting neurons, their entire morphology. So, for example, if we take together the two biggest projects up to date, mouse-light from Genelia Farms and Braindale from the Allen Institute / China, and which are all publicly available, we have more than 3,000 at the moment fully reconstructed long-range projection neurons. So the question is, what can we do with them? Like, can we, for example, use them to better understand how different types of cells connect to one another, thus better understanding the information processing pathways of the brain? We can draw schemata, nice schemata, when it comes to local inhibitory interneurons, but the situation gets way more hairy when we introduce those long-range projecting neurons. So this is how the Neurons United project was born. It's let's say, pan-European initiative. Like we have multiple institutes and universities from all over Europe, starting with Radboud University here in Nijmegen, then having people from the Jülich Research Center in Germany, the CISA Institute and the University of Pavia in Italy, and the Autonomous University of Madrid in Spain. So to summarize, like some basic bullet points, we're interested in generating golden standard reconstructions of single neurons, providing tools for the registration and the visualization of those single neurons, searching for similar ones in the big databases, and last but not least, essentially properly extracting useful statistics to update already existing models, either on the multi-compartmental single neuron level on the level of brain network. And as a use case, we're interested in the cortex, the thalamus, and the cerebellum in connectivity loops. But for the sake of time constraints, I'm going to focus on our work between the cortex and the thalamus. All right. So someone might ask, if you have more than 3,000 fully reconstructed neurons, why do you still want gold standard reconstructions? Well, the point I want to make here is that even in 2022, like those industrious 4D-like approaches that label dozens of neurons together and use semi-automate approaches are prone to registration errors. And registration errors can compromise your result. So I would say that the trained eye of the expert neuroanatomist is still crucial to be able to delineate important neurons. And by that I mean like focal, hypothesis-driven, smaller scale experiments that target a particular nucleus. And you can say that you have your lab, your small lab, and you're able to fully reconstruct, let's say, 20 principal neurons, so that you can understand how the variability of morphologies changes across your nucleus. So one thing is this. Then we have the group of Francisco Clasca from Madrid that essentially have developed like a gold standard approach to effectively label the entire morphology of a single neuron, which utilize its anterograde tracing, which means that, again, there is an injection that is carried through the terminals, and they utilize RNA electroporation to effectively transmit a SynBis virus-expressing enhanced green fluorescent protein. Now, it's section-based, so they utilize coronal or sagittal sections, and 2-photon microscopy to trace the neuron, the entire morphology, and the reconstruction is being used with the Neurolucida software. So that's one thing. Now, as soon as the neuron or neurons have been have been reconstructed, then is where the Tiesenha group from Radboud University comes into play, where they have developed a pipeline to essentially first assist in the reconstruction, but then register the data. So let's take the first step. Well, we can understand that we have now image slices of coronal or sagittal sections, and after tracing, you have essentially scattered axonal fragments. So the first part is essentially the alignment and the stitching of those axonal fragments together. And for that, there is already a link to a tool called the morphology stitcher. So as soon as there is a stitch and the axonal fragments have been essentially assembled together, then you have a fully 3D representation of your neuron in an XML format, for example. Now, when you have this, you can now proceed in going to the raw data and registering them in space. In this case, we have the Allen reference atlas. So you go image slice per image slice, and for each image slice, you essentially first find the closest section from your template that it's in terms of matching, and then you perform a nonlinear transformation to fit the anatomical template. And for this, we utilize two tools very important for neuroanatomy. One of them is very important for eBrains too, Quickly and Elastics. Now, the problem is that those type of images, the contrast is still making it too difficult to register the image in one go, which means that you need a second level of realignment or re-registration, which means that you take this fully 3D representation in XML that I said in step number 1, and you make a point-to-point correspondence to re-register the morphology again, so that it's more effective in the end. And you can also check the morphology viewer tool from Rembrandt that you can utilize to essentially load up in an SWC format morphology and visualize it. But we'll talk about visualization very, very soon. Okay, so here I'm going to talk about something that is a little bit more in progress compared to the first pipeline. Here we're trying to develop a sibling pipeline that instead of a single neuron is focused on registering populations of neurons. So I still believe that populations are still important, but one example of a population that you can utilize is to utilize a BDA tracer, for example, that can target 10 to 100 neurons. This way, you are able to essentially track down the subpopulation that you're interested in instead of infecting a huge area and having something that might be a bit messy. So there is a BDA tracer being utilized by Mario Rubio Teves from the group of Clasca in Madrid. And there are anterograde tracing experiments for those populations. So then the thing is, now because we have a population, and we still consider it quite a difficult project to disentangle axonal tree per axonal tree for each one of the members of the population, we just utilize a way simpler pipeline. We essentially utilize the queuing workflow that is very important in eBrains, namely tools such as Elastic, Quickly, and Visualign. And we do the following steps. First, we segment the population from the background of the image using Elastic. I guess this is quite straightforward. And then we utilize Quickly or Deep Slice to find the nearest section. Now, Deep Slice, I don't know if you're very familiar with it, it's quite a recent tool. It's essentially the deep learning sibling of QuickNI. Instead of utilizing a manual input, it has very powerful deep learning capabilities and does automatically the finding of the nearest section for you. And Visualign relies essentially on user-generated landmarks, and it uses this to make the nonlinear transformation to fit. If you have this, then you can map the segmentation to the 3D template, and then we have the development of cortical flat maps to visualize how the population is being spread across the cortex. Speaking of visualization now, we have developed a web browser essential tool called the Neurons Reunited Viewer that essentially allows you to visualize all the morphologies I talked about before in 3D, but also to upload your own in SWC format, for example, or JSON. The first thing I want to show here is the flat map. This is predominantly based on code from the Allen Institute, but we have made some further improvements, such as, for example, the delineation of the anatomical boundaries and the appearance of the acronyms of its area so that you know where you are. And we have also played with the contrast to make the barrels appear in the barrel cortex. So here, for example, we see how two trees are being spread across different visual areas. And here we have like as a backend, it uses the scalable Brain Atlas Composer, a 3D visualization tool, again, developed by Rembrandt Bakker, who is here. And here you can essentially, this is a 3D tool, so you can load up your morphologies and see how they're being visualized and rotate around the template brain, and so on and so forth. So then you have the obvious question is, what can you do with, besides making pretty plots, what can you do with the morphologies that you visualize together? So here's the idea that I have. Let's say that you're studying a particular nucleus in the brain, let's say the posterior nucleus, and you're working with expert neuroanatomists, and you've managed to track down 10 to 20 neurons. This is still not enough for, let's say, an adequate sample size to make statistics. So there are many ways you could go, right? One would be to clone neurons, but I'm not that expert in cloning. And another would be to essentially utilize, again, smart tracers to infect the population, like the BDA tracer, for example. And the other, I thought, is just having a tool to search for similar neurons in the big, large-scale initiatives. So that's what we do, because we have downloaded and stored all the neurons in some servers from neuroinformatics, and essentially you can load them directly in the Neurons Reunited Viewer, right? So then if you download all the Braindle and the Mouse-like neurons, you can proceed in a comparison. But the question is, how do you compare a neuron with your neuron? It's not so straightforward, because it's not like you have a matrix to matrix or a vector to vector, but rather mathematically you have a three-dimensional point cloud, let's say here the blue one, and another green point cloud. So you also need to find the correspondence between the points comprising the point cloud. For that reason, we have adapted and adopted a technique called the coherent point drift, developed in 2009, which is essentially a probabilistic method employing an expectation maximization strategy, which iteratively tries to solve two problems one after the other. In the expectation step, you want to compute the probability of corresponding 2 points from the green cloud to the blue cloud, using essentially inferring the likelihood that a blue point has been sampled from a probability distribution around the green point cloud, and then you can integrate over all probabilities and now have one-to-one correspondence. And then you can go to the maximization step and solve a Ridges registration problem. Let's say you want to find the most optimal rotation, scaling, and translation, and then you go again step number 2 until convergence, rinse and repeat. Or you can extend it to affine transformation or nonlinear versions, but let's say for the sake of simplicity, we want to solve a rigid version here. So we see here the attempt to register the green neuron to the blue one, and after 10 iterations, there is some relative convergence. And then when it's converged, here, for example, probably it's not very visible, but it's like this light brown. Now the green has become the light brown. It's a bit closer to the blue. We then take a measure like the mean square error and say, okay, this is how close they are. But then you can ask to the system, okay, give me now the top 10 or the top 20 nearest neurons so that I can study them together. All right, so speaking of scaling now, let's say that you have 20 neurons that you have tracked down from your local lab, and for each one you found 10 or 20. So now we're talking about a sample size in the order of magnitude of 100 to 400 neurons. So now we need to start think what kind of statistics can we use for such populations, because now we go to the terrain of mesoscale, of brain areas. So we consider as a case study the ventral posteromedial nucleus of the thalamus, which is like a very simple nucleus with neurons being as clonal to each other as possible. So we want to check what happens if we just put them all together, because with the Maslowite and the Braindle together, we have 280 fully reconstructed VPM neurons, which makes it quite a good sample size in my opinion. So we'll just visualize them together with the neurons reunited viewer. We see this beautiful picture right here, but to me it's still a little bit complicated. So what we did here is we adopted the color coding strategy to label each neuron based on its dominant projection target, the area that it most strongly projects to in terms of axonal length. And then we applied a dimensionality reduction strategy called TSNE to essentially make a scatter plot and see how the data is being distributed in the two most dominant dimensions. And you see that you can, it has like a quadrant-like representation, which is pretty consistent with the color coding. So not all neurons predominantly target the same area. And not only that, but when we check the simultaneously targeted areas, you have 100 neurons that are targeting 2 areas, and 25 neurons approximately that target 3 areas. So it's not as simple as saying that, okay, they all target the barrel cortex, for example. So to go down the rabbit hole a little bit, I made a more simple, at the same time, maybe a more complex analysis, which is I just focused on the somatosensory area, on the sub-areas of the somatosensory cortex, and I tried to create a checkerboard pattern. What I did is that for each of those 280 neurons, I just checked where they have their axonal terminals. So 1, if it terminates in an area, 0 if it doesn't. And then I just group all the unique motifs that I found from the VPM neurons. This creates a checkerboard patterns where you can say, for example, that 35 neurons terminate in the barrel cortex alone. Then you have 44 that terminate in the barrel and the supplemental area, but then 26 of them terminate in the supplemental, not in the barrel, but terminate in the mouth, for example, and so on and so forth. So you could say that you can find 30 unique projection motifs, which is highly in line with what Han et al. Found in 2018 when they were studying single neurons from the visual cortex. So they would find neurons that had dedicated output, projecting only one nearby visual area, and then you had broadcasting ones that bifurcated or trifurcated to nearby areas. And not only that, but Peng et al. That released the seminal BrainTel paper, when they did topography analysis in the VPM neurons, they found some relative topographical organization in the somatosensory, in their terminals in the somatosensory cortex, and some relative topographic correspondence in the VPM, in their somatal locations. So all I'm trying to say here is that you can, even in such a simple nucleus, such as the VPM, you can find subpopulations of neurons that are topographically organized and exhibit higher order connectivity patterns. And this is something that we need to think, especially people interested in neuroanatomy, on how to conceptualize this, because if you utilize classical DTI experiments or bulky tracing experiments, you probably effectively are going to label all of them in one experiment, and therefore you're not going to be able to dissect those complexities. So it's something that maybe we can utilize, we can take advantage of this new trend of single neuron reconstructions and try to uncover such statistics as I'm interested in, for example. So last but not least, someone might ask, okay, but what is the utility of all this? So I am able, let's say, to highlight a number of statistical patterns or motifs, and then we would have to test them somehow. And this is what we're interested in. I would say that I would like to act as a mediator between the experimental world, where we can extract essential morphological reconstructions, but also between the modeling people, to which we can provide useful statistics to serve as biologically plausible constraints to already existing models. So one example is a basic somatosensory thalamocortical model, which we want to test. So it's a very simple model that has been developed by the group of Sasa van Albada from Jülich, mainly for 2 of her students, Hanjia Jiang and Renan Shimura. So what they did is they take the classical Podjans-Disman model, which is essentially a barrel column, and they adapted it. So from the Podjans-Disman, in case you're not familiar, you take a column and you can break it down into 4 distinct layers. And within each layer, you can have a population representing a cell type, right? One for excitatory and one for inhibitory. And each population is essentially a simple compartmental leaky integrate and fire neuronal model, all right? So then you can connect them intralamellarly or interlamellarly, each population with different connection probabilities. So the extension here is to break down the inhibitory population into somatostatin, parvalbumin, and vasoactive intestinal peptide, and introduce 2 additional modules. One module for the reticular nucleus and 1 module for VPM. Now the way that everybody is interacting with is by connection probabilities that, for example, here, they have been estimated based on the statistics that I talked about before, that we extract from the morphologies. So there is a forward feedback interaction between the VPM and the barrel column, and the reticular nucleus interacts with the VPM and acts as a kind of inhibitory source. And okay, so 200 reticular and 200 VPM neurons, for example, here are simulated. 6,500 approximately cells in the barrel model. And like things that, this is a very embryonic stage. Things that we would like to try is, first of all, check this in a spontaneity, if it can reproduce spontaneous activity better than when defining, for example, statistics based on the exponential distance rule by updating it with morphologies. And then, of course, beyond spontaneity, someone might ask that if it will be able to reproduce experimental measurements, but I guess that will require some time. And last but not least, we're interested in scientific integrity and transparency, so we would like to upload our data and our pipelines, as well as the related documentation in the form of Jupyter notebooks in the eBrain collaboratory, as well as other publicly available repositories, such as Donders repository, GitHub, and the Scalable Brain Atlas that I mentioned before. Two examples here. The dataset from the latest publication of our collaborators in Madrid has already been uploaded to the eBrain's collaboratory, and our pipeline for registering in 3D space single neuron reconstructions has already been uploaded. I would say that this work would have been utterly impossible without the collaboration of so many wonderful minds, which I would like to deeply thank. And therefore, I conclude my presentation, and I want to thank you for your attention.